Hey everyone, welcome today. I'm Nate O'Connor. I'm a scientist and product manager here at MBF Bioscience. Today I'm honored to have renowned neuroanatomist Dr. Chip Gerfen with me. He's just getting connected now, but we're going to be presenting a workflow we've developed for mapping neuroanatomical structures that have been labeled in histological sections to a whole brain standardized coordinate system. So here, that's going to be the Allen CCF and its corresponding atlas. That you'll see that lets us compare measures across animals, groups, experiments, and even laboratories. First, Chip is going to give some scientific context, and then we're going to run through the specifics of the workflow that we've developed in our NeuroInfo. So today's webinar is going to be an outline of the basics of our workflow. And provide some context. We've already got follow-up webinars that we're scheduling that will go into specific details of workflow steps. So at the end, we're going to have a Q&A session, and so be sure to use your GoToWebinar interface to ask us questions. We'll be watching. One other thing, COVID related, and basically everyone at MBF and Chip, we're all remote. So I wanted to make sure that some things that can happen when you're remote didn't interrupt us. So parts of this are going to be pre-recorded and played, but we are here live and we're watching for questions, and the Q&A at the end will definitely be live as well. And there's also going to be a replay link that's available too. So our technical guru is our marketing manager, Pasang Sherpa. She's in the background overseeing things, so we're all here. Pasang, I think you can start things now.
My name's Chip Gerfen. I'm a neuroanatomist in Bethesda at National Institute of Mental Health. And today I wanted to show how we use the NeuroInfo platform to be able to make reconstructions, whole brain reconstructions from coronal sections, and then register those to the Allen Atlas. The Allen Institute, and led by Honghui Zhang and Lydia Ng and Julie Harris, really pioneered the ability to register brains from different brains into the common coordinate framework. And this was the mesoscale connectome of the mouse brain paper that they published in Nature some years ago. Now, the major advantage of this was that they took individual brains that were coronal sections, and you can see this is one of the brains that they did, and they made injections, AAV viral injections into the cortex, and mapped the projections through the whole brain using, in this case, and did every section, and then they registered these sections to a common atlas. So the advantage of this is that it allows for taking different data from different animals and quantifying and looking at relationships at one, very easily in a quantitative manner. So in this case, I want to show that these are injections from 5 different brains into different parts of the somatosensory cortex, into the mouth area, the upper limb area, the lower limb area, and 2 parts of the barrel field. So these are all taken from different animals, registered to their common atlas, and then we can combine them into that atlas space, and then view and look at the topography or different the way that these projections from the cortex project through the different parts of the brain. So these are the 5 injection where the 5 injections are in the cortex, and then this is a sagittal view. You can see the injection sites in the cortex, and then the projections going through the striatum into the thalamus, and then coronal sections. We can reconstruct these and see the relationships between the projections from different areas. So one of the things that this allows is to be able to see that the organization in the cortex is somatotopically organized, so that the relationship between the different body parts is maintained in the cortex. We can see that those projections into different parts of the brain are also maintained. So we can see the somatotopy. this has been known for a long time, but the ability to look at this in the same coordinate space allows quantification of the relationship. So here we can see that there's topography in both or somatotopy, the relationship in the cortex is maintained in the projections into the striatum, and also maintained in the projections to the thalamus and even to the pons. So this enables being able to actually do quantify the relationships of different cortical areas and their connectivity. And so this is showing a top-down view of several hundred injections into different areas of the cortex. These are the top-down views of those. And one of the things that could be done would be cluster analysis to compare and analyze the relationships between the different cortical areas and their connectivity together. So this really, this approach, in my view, as a long-standing anatomist, really revolutionized the ability to quantify and do quantification of the connections within the brain. However, one of the downsides is that the Allen Institute is quite a, has quite resources and a number of people, and it's a large, it was a large study that involved many people to do. What we'd like to do is to be able to do this type of analysis within our own labs, and the NeuroInfo platform enables that. One of the example that I'm going to show today is from a study that was done with Alexandra Nelson's lab, Ali Girsal, Matt Ryan, and from the University of California in San Francisco. In this case, they study the neurons in the striatum, that are responsible for L-DOPA-induced dyskinesia in an animal model of Parkinson's disease. The study that I want to show will use cases from their study in which rabies virus was used to, was injected into the striatum, and retrogradely labeled neurons that express the D1 receptor. And so it transsynaptically labeled the neurons in the striatum that project to the from the cortex to project into the striatum to D1 neurons. And so this is the rabies injection. You can see the transsynaptically labeled cortical neurons. The NeuroInfo program then automatically uses cell detection methods to detect the cells in the cortex. Those cells are then, you can see them in the distributed in the cortex, and also the injection site. And then the location of these cells is then mapped into the Allen coordinate framework. And that framework then enables us to count or look at the number of neurons in different, not only in different cortical areas, but in the different layers of the cortex. And also in the different layers of the cortex, from the front of the brain to the posterior, also in regions such as the globus pallidus and the perivesicular thalamic nucleus. So now I want to go through and show how we can do that using the NeuroInfo program.
I just wanted to thank you for that overview, Chip. It really showcases kind of the experimental case and a workflow that you can implement with the platform. So I'm here, Chip. Refer to me anytime you need any questions, and I'll be reading questions from the audience as well. Just wanted to chime in, let folks know I'm here.
Okay. Thanks, Nate. So, okay, so the first part of the process is that we have to take our coronal sections and reconstruct them into a 3D volume. And this is done using the BrainMaker workflow in NeuroInfo. So this is located in the top, in the panel up here in the upper left. So we click on that. So this provides the workflow that we use to reconstruct. So what we've done, what we do is we take our brain in the sections, and the first thing we have to do is to add the slides that we're going to use. And in this case, I'm just going to use a brain just to demonstrate how this works. So normally I would load in all of the slides from a particular brain. In this case, I'm just going to load one in just for the purposes of demonstrating how it works. So these are sections that are serially mounted, through the middle of the brain. Normally we would load in all, for a particular brain, for a particular mouse brain, there's typically about 7 to 10 slides fit all the sections on a particular mouse brain. So in this case, we're just loading in one slide, and they're in sequential order. And so the next thing we do is to go to the next step. So there's two different ways that one can have slides mounted. You can either have them sequential, as I just showed, or staggered, which is what we typically do. In another webinar, I can describe the details of how to do that. But for the purposes of this, I'll just show a sequential mapping, or use sections that are sequential. And in this case, there's a variable number of sections per slide. Go to the next step. So what it's done now is it's loaded in all of the images from that particular slide, and it's outlined them. So you can see a red outline around all of them. And it's done this for all of the sections in this on this slide. Again, for the purposes of demonstration, I'm going to remove most of them. So I can select them, and there's a variety of things that can be done here. Sometimes if the sections are overlapping or things, you can modify the outlines here. But for this purpose, I'm just going to delete that. We've got 4 sections left. Next thing we do is to have them ordered. It automatically orders them. So this is section 1 on the slide, section 2, section 3, section 4. If I'd done the whole thing, it would have done section 5, section 6, all the way through to the end. And then when I went to the next slide, it would begin at the next slide and go on. So go into the next step.
And so, Chip, these were imaged at what magnification? What's the resolution of these sections?
These are all sections that were imaged at 10x using a 10x objective. Almost all of the things that we do are, from this brain were imaged at with a 10x objective, which is typically pretty good.
And so that's sufficient for like cell detection as well as the connectivity analysis you were showing us.
Yeah, exactly. Okay, so then it's going to go through and do it an alignment. Excuse me. And there we can various ways we can do this. We'll try the fastest first. And this is just for the purposes of demonstrating how this works. Now, so it's going through and it's taking those 4 sections that that have been outlined, and it's doing a simple alignment of them together. And now it's done. We go to the next step. Now it's going to show each of those sections that have been aligned in the window. And this is the first section, and we can register or line it up. We can use a grid, vary the size of the grid. We want to use the grid to get the sections to be somewhat, perpendicular to the axes. We go to the next section. And so now it's overlaying the second section with the first section. And this is the second section, this is the first section, and this is them overlaid so we can see them, and we can rotate them to do minor adjustments, move them about. Want to line up the ventricles. Go to the next section.
So Chip, as you're doing this, is there, I think we found your sections are spaced at what, 80 microns apart?
These sections are actually all done at 50 microns.
They're at 50 microns.
Yep.
So
these are 50 micron sections in there, and go one to the next.
I think I've noticed that this alignment is possible for separations of probably, what do you think is an upper limit, 150 microns between them?
Yeah, we never have gone that high, but I'm sure that should be fine.
I'm just thinking of data I've seen from other laboratories too.
Okay. Anyway, so we go through this, and it goes through all 4, and we line them all up, and then this particular animal has like 120 sections. We would go through that whole process doing the registration like that. And then we go into the next step, and it allows us to save that all of the sections that have now been aligned with each other into a large stack, a JPX stack. And so we would save that reconstruction, save it to a file, and that's what we use to for the next step.
And that format, Chip, allows you to have compression without losing features or a change in cell detection performance.
Yeah, so there's a number of variables. We can change the compression. We can actually also, we could save it rather than as a JPX stack, which is JPEG 2000. We could save them as TIFF stacks, or save them and save the individual sections, save the channels separately. It's quite a bit in the future webinar when we go through this. I can go through all the details of that, but there's quite a bit of variability of how we do this. Okay, so that's just to demonstrate how the BrainMaker part of this works. I'm going to stop the workflow. Actually going to cancel this, close all these files, and I'm going to open up another. This is going to be a case that we've already registered. I mean, it's been aligned with itself, and we've marked cells, marked contours in this, and all. I'm going to hide those for the time being, and we're going to hide the markers, and the rabies injection site. And fiducial markers. So now I've loaded one of the brains from it's been injected with rabies, and it's been we made a 3D volume with this, and I'm going to arrange the windows. So in NeuroInfo, there's various windows that can be opened that allow functions to be applied to the images and to the stack and all. One is the orthogonal view, which allows us to go through the go from rostral to caudal through the brain. Then we can also adjust the various colors. Enlarge. So this is a brain that's had an injection of rabies into the striatum, and it's transsynaptically labeling neurons in the cortex that projected those D1 neurons in the striatum and march through this brain. And you can see through the orthogonal view that you can see as it's marching, the section that it's at. There's sections there are neurons in the globus pallidus, neurons back in the thalamus. They're also labeled. So one thing that we do for registration is, and this is something that we'll talk about later that we can do, but we put in fiducial markers, and these fiducial markers Put them here so that we outline the outer, outer part of the cortex, the white matter that delineates the striatum, structures, showing the globus pallidus. We can use these markers, these fiducial markers here, to be able to later go in and quantify or see how accurate the registration is to the Allen space. So this is again a whole, the coronal sections have been reconstructed into a 3D volume.
So Chip, just a question. These fiducials are so that you can get a measure of accuracy. They're not required for the registry.
Not at all. Yeah. I just like to use them because we go through and we want to see how accurate the registration is to the Allen space, and these help.
And how, once they're cast or matched into the Allen space based on the image matching to the Allen space, what's a typical accuracy that you're seeing out of this matching process?
We typically get less than 100 microns. So it's between usually about 80 microns accuracy. And considering that neurons are some 25 microns in diameter typically, that's about 4 neurons. And that's I think that's fairly accurate. Okay, I'm going to hide these fiducial markers. So first thing we want to do in this, or we do in these brains, is we want to count the neurons, or we want to mark the neurons that are labeled. So this is a going to turn off the Nissl image and the other, and these are the neurons in the cortex. Navigate. See that this is section 36 out of 120 in this particular animal. So to count the sections, or to mark these, count the sections, but to mark the neurons, we use the cell detection workflow. And this has a number of different steps. So the first thing is we're going to detect cells in 2D. There's the option of detecting doing 3D cell detection, but the tissue that we have now is not suitable for that. And
that might be suitable for something like cleared tissue whole mouse brain imaging or something, right?
Yeah, exactly. And or even confocal images and all.
Right.
So the first step is it goes through and we're going to, just for demonstration purposes, I'm just going to detect the cells in a single plane, and it's going to detect through the whole. It could detect just in the image that's being shown, but I'm going to do it on the whole section. And these are want to detect the red cells. If we had other cells with other other labels, green cells, we could detect them, but we're going to detect red cells. And then the way this works is it uses the Laplacian of Gaussian algorithm for cell detection, pretty standard algorithm that's used. And so there's 3 main parameters that having to do with the size of the cells that are the target of what we're trying to detect. So we can actually go in cells, find the largest cell, and it's around 20 microns, and then find the smallest size cell. And these are not, they don't have to be super accurate, these measurements, but somewhere in the ballpark. So typically find that where you can use parameters of 25 and 10 are fine, and then run a preview, and it marked the cells. So one of the things that we see here is that a number of dendrites are marked, and we can adjust the threshold. And so now we've adjusted it so that it's eliminated most of the dendrites that are being labeled without while still counting the neurons. We've also there's adopted this or developed a machine learning algorithm that's going to be that can also be used, and the machine learning is great. The neural network that's developed out of that is great because it actually eliminates the detection of dendrites when we're looking for pyramidal neurons. Anyway, so we go to the next step. One of the things that the way that the Gaussian of Gaussian works is that it detects objects that it detects, and then it assigns each of those objects a strength value, and we can actually adjust the strength value, and here you can see the histogram. So most of the objects that it detects are actually in the background, or the dendrites and all of that. So we can adjust the strength value. And then after we're satisfied that that it's accurate, we hit detect, and it will detect all of the neurons in this section. Not only in this section, but we can have it detect all of the neurons throughout the brain. So I'm going to close the workflow. So this is a brain that we've processed before, and I've gone through, and I'm gonna unhide the neurons that are marked. Change the color. So so there's some, for the most part, it's done a pretty good job of detecting all the neurons. Going to turn these back to red. There's a lot of things we can do. Okay, so now we can visualize this in 3D. So we can view this brain in 3D, and so you can see all the cells that are marked and all. So now what we want to do is to register this 3D volume into the Allen space. Close the 3D window. So we have our 3D volume that's been reconstructed and with the cells detected. And now we want to do section registration. And the section registration, the first part of the section registration requires an atlas calibration, which just is a simple thing that adjusts the dimensions of the experimental sections to the Allen space. And so the Allen space, and it's dependent, everybody cuts brains. It depends on what tool, what method is used to cut the brains. And some methods compress, in the dorsal ventral dimension more than they do in laterally, and so the atlas calibration allows for that adjustment. Once that's been done and we've established that, then we use the section registration workflow.
And that calibration usually occurs what, like once per protocol, once per brain?
So or actually, we use the same atlas calibration all the time. we've done it for 1,000 brains. So
you really only have to do it once.
Yeah. And the other thing it does is to establish what channel is being registered. In this case, the sections that we always stain sections with a fluorescent Nissl stain, and that's the channel that's going to be registered. Okay, the section registration opens up two windows. One is a window that shows the experimental section overlaying with the Allen space, and the Allen space in this case is green. We can either use grayscale or color. I like to use color, and then we can vary the strength of the underlying experimental section which is shown in red. Transparency, we can go back and forth. This is the Allen and the experimental. Okay, so the second window that's open is actually contains the controls for registering the sections into the space, into the Allen space. On the right down here is showing the Allen space in sagittal view, and this is actually not, even though this is the Allen space, we've registered the Allen space once to our an average Nissl brain, and so that's what this is, and it accomplishes the same thing. That's the space is the same. Okay, so what we do is we go to a place in the middle of the brain. So I'm navigating to, I'll describe why we do this in a second. Okay, so this is a section through the middle of the, somewhat middle of the brain, and the reason I show this is that various structures, depending on where they're located in the brain, you can tell where they sit in the rostrocaudal axis. So let's. So this is the. Rotate that space. Go from the front of the brain to the back of the brain, and you can see that things like the location of where the hippocampus is in a coronal section. You can tell where you are in the rostrocaudal axis by its position. So as we go caudal, you can see that the hippocampus starts to rotate around, as we go caudal. So you can watch the rostral and other fiber tracts like the anterior commissure as it crosses. One can tell where you are in the rostrocaudal axis depending on the location of various fiber tracts. Other markers. So you can also look at another one that's good, is you can tell where the basolateral, the amygdala is a good structure that can tell where we are in the brain, where we are in the rostrocaudal axis, depending on the size and shape of that, or the fasciculus retroflexus. Point is that these different structures in the brain give us an indication of where we are in the rostrocaudal axis. So the first thing that we do is to try to move the Allen space, move where we are in the Allen space to be close to where the experimental section is. So you can see I moved it so that the hippocampus has a similar shape. The mammillothalamic tracts are just above the ventricle. The optic tracts are coming in there, and the amygdala is here. So in an ideal world, we would always cut our brains exactly perpendicular to what the Allen space is. So this is similar to when you look at an atlas, at a paper atlas, and you want to locate where you are in the brain, you look and say, okay, well, I'm here in the brain for the hippocampus, but the section that I'm looking at in the atlas for the paraventricular nucleus or the basolateral nucleus of the amygdala might be in a different section, and that's because we don't cut our brains, or the brains are not always cut exactly perpendicular to the Allen space, and so we can. So
you could end up between pages of the atlas.
Yeah. So what we would, the purpose is we want to find where this section, this experimental section, where that lines up with all of, within the Allen space. And so we want to find that, find that position. That's what registering to the Allen space, or registering to any common coordinate space is. We want to see, where is this space exactly? Where is the position in the experimental brain? Where is it exactly in the Allen space? So this is the mammillothalamic tract in the Allen space. You can see it sometimes if I scroll over it. You can see that it names it in the lower left. And so I want to find, here it's not lined up properly. So what we want to do is to find the space in the Allen space that exactly corresponds to this section. And so we do that by a couple of things. One is that we can move this so we can see an overlap, and now we're going to use the adjustments. And what we're doing is we're adjusting the Allen space. You can change the scale. When I talked before about the scale, even though we've calibrated the atlas to that, we can make adjustments in individual cases, and then we can shift and line them up. So you can see now that see where the hippocampus is, and try to line up the hippocampus properly. And we also want to look at the mammillothalamic tract. Let me just move this a little bit more. So you can see I'm moving these bars to do the alignment, and then sometimes we need to rotate. So you can see now I'm changing. So now this, where we are in the Allen space, this section is very different than where it is ventrally, by adjusting this rotation. So we want to find a place where we can, where the hippocampus is, the right shape, and also where the mammillothalamic tracts and the amygdala is correct.
And so you can see now that angulation that you're kind of approaching, and that's the effect of not being able to cut in canonical coordinate systems, right?
Right, exactly. So sometimes also there's the right side of the brain in this case is a little bit caudal to the left side of the brain, and so not only can we rotate in this axis, but we can also rotate a little bit. You can see. So this cut then shows what the cut is, the angles through the Allen space to give the best registration of this section within that location. So pretty satisfied with that. We would go through and make that the reference section. So the idea is that the principle behind this registration of the whole brain is that, well, let's back up a second. So one thing we could do is say, oh, okay, now we could, if we wanted to, just say we would just want to register this single section into the Allen space. And so now when I scroll over the experimental section, I see that in fact it's lined up the basolateral amygdala. The basolateral amygdala is, is perfectly lined up. The habenula, hippocampus, all of those things. So they're all fairly, so we could do, if we wanted to just do a single section, this is, we can then map this to the atlas and get the coordinates of our neurons that we've marked before in the coordinates in the Allen space. However, what we typically would like to do, or what we like to do, is to be able to register all of the sections into the Allen space. And so the principle that we use is that this, the fact that our sections are not perfectly orthogonal to the Allen space, however, they should be consistently non-orthogonal. And that's because when we cut the brains, we don't cut at different angles, from section to section, that the knife is in a fixed position relative to the brain. And so if we can find the optimal offsets in the rostrocaudal and dorsal ventral and right left dimensions, these offsets, if we can find what are the optimal ones for this particular brain, for this section, we can apply that to the whole brain and then register all of the sections in the brain using these offsets. So one thing we can do is to go and test how good that offset is in other areas. So I'm going to go rostral and see. So these are the offsets. This is the offset. I'll go rostral. And even though it's offset in the dorsal ventral axis, these offsets can shift the so we can see that the anterior commissures in these locations are accurately mapped. So this suggests that, in fact, that offset that we used from the middle part of the brain actually can be applied throughout the whole brain. We can go through and test that. Once we've established that we're satisfied that this offset is going to give us an accurate mapping, then we use that and go through the march through the whole brain and have it do the registration. So when we first did this, and we said we looked and said that, or looked and saw that that it was displaced in the dorsal ventral axis, the program actually makes that adjustment. So it makes the adjustment, this dorsal ventral offset is actually made. I'm doing it here manually, but the program, when we go through it, it does that. So once we've then established that this section 57, which was the section that we were at before, where we did the establish the
So Chip, you're basically finding an equivalent section in the Allen space to your experimental section, and then using those offsets to kind of inform the next transforms for all the other sections.
Right, exactly.
Gotcha.
So we want to do is, and so that's what we call the reference section. So the reference section is then the section is going to those offsets, those transforms are going to be applied, just going through to see how well it's working out, and it's doing pretty good. So it's just marching through now, section to section, and all. And this is just what I do is to make sure that the offset I've applied is going to be appropriate through other sections. I mean, through the whole brain. Again, I'm using this orthogonal viewer to okay, so once there, so now what I do is that I go and I say, okay, I want to apply that offset to all of the sections of the whole brain, and I'll do that. Although for the purposes of what we're doing, I'm just going to have it run through a few of them. Let's go like section 55 to section 16, and it's going to lock the sectioning angle. So that it's going to lock those angles, the angle that was originally that we found for that reference section, and it's also going to lock the relative scale, and then we run linear. So what it's doing is it's now going to go to each of the other sections. Now I only set it up so it's going to go 55 to 60. If I'd hit all, it would have gone from the front of the brain to the back of the brain. So what it's doing now is it's going to each of the sections in sequence, and it's finding the match of the best match of using these transforms that are the these angle transforms at those sections, and it's applying those transforms are then being applied to all of the cells that have been marked in that particular section. So it goes through this, and then it'll so these are just the first step is that it applies a linear transform. What that basically does is it's just applying linear, the angles, dorsal ventral and right left angles that those offsets that we've determined, and it's just a linear transform, finding the best linear match. So the next step after we've done this with this linear transform, then we're going to go through and we're going to run a nonlinear transform. And what that does is you can see that there's like the edge of the cortex isn't always well matched with the actual edge of the cortex in the experimental brain versus the Allen space, and so the nonlinear transforms then make minor adjustments to each of these sections to better fit the Allen space.
And do you typically always use the nonlinear transform after the linear transform?
Yes.
Cool.
Because the brains are so it'll fill it. Again, this is a good example where it's just not matching exactly with the. That's because and it can't, because all it's doing is it's applying a linear transform, and so the nonlinear allows for some better warping.
Right.
So after it does that, then we would run nonlinear, and then that's all done, and it'll go through. And so depending on how fast your computer is, this process can take. Then also depending on how many sections you have, it can take half an hour to several hours, depending on how fast your computer is. But then it does it all automatically, and there's nothing to do. When it's all finished, then we hit map data to Atlas. And what that does is it maps all of the markers that we put into the Allen space, and it gives and we have options. You can determine if you want to do it through the whole brain, in this case the 120 sections, and it gives a marker summary, the points, and also includes the images. And we say begin, and it would do it. But I'm going to cancel because that actually doesn't take it takes typically about 5 minutes.
So that functionality takes and it applies the transforms that you found for the images into the Allen space.
Exactly.
To any marker coordinates that you've already placed with the cell detection.
Right. So what that does, what we've done now is we've taken that all of the markers that have coordinates in the original image, and all of those markers those coordinates now, instead of being in the original image coordinates, are now in the Allen coordinate space.
Gotcha.
So so anyway, and that's done through map data to Atlas. So what this is what I've just done is this is the file, and then it generates several other files, and it generates a summary file which contains all of the it contains the data, the number of counts for each of the structures in the Allen space. So we've got different markers in this. It had the cortical labeling of the cortex, and so it gives the number of counts in all of the structures that are listed in the Allen coordinate space. So there's some 2,400 or something like that. Anyway, and it breaks them down according to the area, all of the neurons in the cortex, or in the different cortical areas, somatomotor areas, primary motor, secondary motor, and also by layers.
And by hemisphere, right?
And by hemisphere. And then it also gives a file that we can actually visualize this. Close all of the original images. It gives an output file. So this is the output file, and initially it starts at the front of the brain, so there's nothing to see because there's nothing there. I'm going to unhide the markers. We can see all of the markers through the, change those to red.
And Chip, you can actually see in those red markers in the orthogonal view, you can see that angle throughout the extent of the, from the front of the brain to the back, in where those planes were projected via that offset that you found.
Yeah, exactly. So you can see that after we've done the linear transform, so what this top-down view is showing is it's showing the location of the markers now in the Allen space. And so if the brains were perfectly orthogonal, these would be straight lines because each section, the markers for individual sections are in the same plane. However, because of the offsets that we've used, there's some angles. You can see that there's a little bit of an angle from the right to left. And also, if we put this in the other dimension, you'd see the angle in the top to bottom.
Right.
So now we're going to navigate to, we just want to see the markers in an individual plane. So now the Allen space is then shown in three different ways. One is that there's a grayscale image of the Allen atlas itself, which is what's being shown now. It's also the colored version of the Allen space. And then there's also, this is the two photon. This is the 2-photon image.
So that's the original average of the 2-photon block face mice.
Exactly. And so we can see here that how well the fiducial markers, how well they line up.
So now we're actually looking, Chip, at all the markers that you placed or that were automatically detected transform with their coordinates transformed into the CCF, or the Allen average mouse brain atlas space.
Exactly. And so then we use these fiducial markers to show, how well they lined up. This is the edge of the and let's go. So this is in the so sometimes the fiducial markers actually gives an indication that, and if these are too far off, then we go back and redo our registration. But for the most part, they're pretty good. And so this is the injection site. These are the neurons in the layer 5 of the cortex.
Cool.
Okay, so we can go through and we can see the, you know, how well these are all mapped now into the Allen space. And let's see what the 3D looks like. I'm going to show that later. You know what we're going to do to show that? Okay, so we've done that, and now I'm going to open up. So anyway, that's how we go through, and each of these, I went through pretty fast the different parts of the program. There's a lot of, you know, other, you know, to be able to actually run through this is a lot of other things that need to be, you know, learned and all of that. But that's, but I thought this was just to give an overview of how the program can actually generate from coronal sections, a 3D volume of cells, you know, of labeled cells that can then be registered into the Allen coordinate framework. Thank you.
Thanks, Chip. That's fantastic. I really thank you for your time today. And this is how you're actually doing things in your lab, right?
Right.
And just as you mentioned earlier, a bit of a time frame, but what do you think of I guess as an end of the day question, how many brains can you get through in a day, or how long does a mouse brain, for the extent, rostral caudal extent that you look at, take you?
So the way we always do this is that to cut a brain, we cut frozen brains, and it takes about half an hour to cut a brain. And then the immunohistochemistry that we run on these, that's overnight, but it doesn't take any, you know, it's not manual time, it's just, you know, waiting time. Then it takes about an hour to, well, 45 minutes to an hour to mount the brain. So the manual part of processing a single mouse brain is, you know, in an hour of cutting and an hour of mounting at the end. Then the imaging is really the key thing that takes the longest amount of time. And that depends on what sort of resolution and how many markers you have and how strong the markers are. So that when we image these brains, and we typically have 120 sections per brain, that's from the rostral pole through the brain stem. And if we have four fluorescent markers, we always image at 10x, which is sufficient to be able to count, you know, it gives fairly high resolution for counting, being able to detect neurons and see processes and things like that. And, you know, for standard fluorescent imaging, it takes about, you know, 3 to 4 hours on our Zeiss microscope. It takes 3 or 4 hours to image an individual slide. And we have 10 slides per brain. Okay. So the real slow part of this process is the imaging, and that takes, you know, as I said, 3 to 4 hours times 10. But that's all automated. And then the processing that we do in NeuroInfo takes about, for the BrainMaker part, where we do the registration and all, that's typically takes about an hour. Everything seems to be about an hour, but it's literally about an hour of you know, going through the process. And then the registration or the cell detection takes less manual time, but it takes about, it can take 3 or 4 hours to actually do the to do the automated cell detection through the whole brain. The brain registration itself, the manual part of it, where you actually have to do something to while you're at the computer, can take, you know, that actually goes pretty fast, about half an hour or something like that. And then the registration itself takes about, you know, between an hour to 2 hours. So if you add all of that up, what we typically can do with this is we can process, well, you know, we could process many brains you doing just the NeuroInfo part. The real slow part is the imaging.
Gotcha. So that I think that's a really a practical point. I'm glad you talked to.
Yeah, I think that we typically, the way we have two imaging microscopes that we run, and so we typically can process from from brain to finish 4 to 5 brains a week.
Oh, cool. Great.
And that's that's basically one person's work.
Gotcha.
And it's not full time, but it's most of the time.
So
Okay.
I think we'll answer some questions now. Chip, thanks so much again for that. Really informative and useful. And with that, we'll take a look at our question panels here. Yeah, that was great, Chip. We've had a lot of questions coming in. I've actually been trying to keep up with them as they come in. Again, we will get to we're at the hour, but I think we can try to answer at least a couple questions. If you have to go, just keep in mind that this will be available for replay. A link will be going out on that. We also will answer all questions that we've gotten in email to you too. So this one question is a really great one. Someone asks, would love to know if it is possible to superimpose expression patterns throughout the brain from a number of reporter lines, basically overlaying them to see where they are the same and where they differ. Chip, I'm going to throw that one to you.
Can you hear me?
Yep, we can hear you. A-okay, Chip.
Okay, great. This is Chip Gerfen. Anyway, I'm a little embarrassed having to listen to that. It's that presentation that was done some time ago. The one thing I want to emphasize is that I went to try to go through this. This is a very powerful program, and I wanted to just give an overview of all the different steps. Each of those steps has a lot of, would require a lot of detail to teach, how to do it, and we'll be providing follow-up webinars to describe, provide that detail. One of the things that we want to emphasize is that the output of this program, what it does is it provides the coordinates for cells that have been marked, neurons that have been marked in a brain, and then it puts them in a table in a format that has the coordinates in the Allen space. So we can take, and we typically can do this, is you can take data from the power of this whole technique is that you can take data from different brains, and because all of the data is then put into the same common space, you can compare labeling or compare the distribution of neurons from different brains in the same space. And so we can do that in NeuroInfo by taking just basically the output of that, when you do the map data to Atlas, take the output of that output file and combine them into a single file, and you can display them at the same time in that space. Or we can output that same data into people that are used to using Python or MATLAB or Fiji or other programs. You can also take that data and put it into to compare. So the answer is yes, you can take data from different animals and compare them directly.
And Chip, toward that, I just throw in there the output of this, because another person has a very similar question. We make available upon request and are publishing our XML format for all of our measurement storage. So you can actually get in there in Python or MATLAB and get at all those coordinates as well, like you were saying. But we have a standardized format for that that's freely available to folks.
Right. Well, it also just comes out in an Excel spreadsheet.
That's
so
right. That's
straightforward.
Another question I have here is what kind of hardware, software setups are recommended, if any? The minimum specs are at our NeuroInfo website. But the other thing I'd say with that is, I'll tell you the rig I'm using here at home. I have an MSI laptop with an i7. It has 16 gig of RAM and an NVIDIA GeForce RTX 2060 card with 6 gigabytes of RAM. And the reason that works is like Chip showed, we're doing things on a section by section basis rather than the entire volume at once. And so you don't really need, a huge computer to be able to do that. But again, the minimum specs are on the website. We've had Chip. I think. Do you want to do Chip one more question, because we're 6 minutes over. One interesting question is what about other brain atlases? This was all on the Allen today, and so I've had a lot of questions about different species as well as different atlases like Paxinos and Franklin. And Chip, maybe we both can answer a little bit on this, but I'll let you go first.
Yeah, so the advantage of using the Allen space is that they've actually annotated the atlas, and so the coordinates and the images are all registered to each other. So when we do the registration to that atlas, then it also can the coordinate space, the coordinates that are provided in the Allen atlas and that common coordinate framework are pegged to structures. And so that's a major advantage. I'm not familiar whether or not the other atlases actually are similar. If they are, then it would be fairly straightforward to adapt them and incorporate them into NeuroInfo.
Right, and that's one of the things we found is the reason it was so easy and the first one we grabbed was the Allen is because they actually have an API and everything is published and really well formatted. And so we worked with them to come up with a way to get that integrated into our software. But it was basically just grabbing some volumes, their ontology and their regions, the segmented regions that Chip showed, and putting those in our software. So it really depends on if these other atlases have that available. Another thing that we will be making available is if you have a digital atlas, how do you get it to use into NeuroInfo? Because it's really just a series of images, XML files, and then an ontology in a CSV file. So we can make that available to people too if they do have their own digital resources for other species. It would be great to work on that. I think we're over, so I do want to end things, but I really appreciate your time, Chip. This was wonderful, and thanks to everyone for being here with us. It was really fun. And we will be having follow-on webinars, so look for emails on that. And we do answer questions, so everything I couldn't get to, we will answer. There's also a forum. There's a link to that right in front of you. And then NeuroInfo is free to try, so there's a link here for that too, and that'll help you start the process of that. So feel free to contact us. And again, this replay will be available in the next week ago. And thanks, everyone. Stay safe and productive.
Thank you, and I'm happy for anybody to contact me as well.
Great. Thanks again, Chip.
Okay, bye.
Bye, everybody. Bye for now.